Hello, everyone. Welcome to the scientific briefing session for before lunch. My name is Lei Shi, and I am currently responsible for the R&D activities at the Biointron Biological. And today I'm going to introduce my topic is going to be Simplified and Humanized, empowering human antibody discovery with AbDrop solution. So let me give you a very brief introduction of the antibody discovery platforms and the major approaches for discovery of therapeutic antibodies has been really matured over the years, and we can basically divide these into the hybridoma approach in vivo with the in vivo feed maturation versus the phage display approach with the various renditions of display technologies. And over the past decades, these technologies have evolved to become more sophisticated and to meet the demand of our scientists to discover the molecule quicker and more efficiently. Therefore, the Humanized mouse platform has contributed a lot to the hybridoma platform. Even more, the single B-cell screening technology has greatly accelerated the discovery process of antibodies. And the advantage of single B-cell technology is pretty obvious in my view. It is rapid, and you can get the cells, B-cells sorted in days, if not hours, compared to months and weeks for hybridoma and the phage display. The diversity you can get from B-cell, single B-cell sorting is also higher with hybridoma is limited by the number of plates that you're actually screening. The affinity is as high as you can get, similar to hybridoma platform. Phage display, on the other hand, there's a various, depending on the technology you use, and the affinity can be varied. The throughput is obviously much higher. We can sort millions of cells versus hybridoma in the tens of thousands. The pairing is as natural as it could be, as we could do find in the hybridoma platform. Phage display, on the other hand, you get a scrambled, recombined VH and VL, although some people may argue this actually increases the diversity of that. So at the Biointron, we are actually focusing on using single B-cell technology to accelerate the antibody discovery process from immunization to validated antibody leads in two to 3 months. That is faster than the hybridoma screening that conventionally used. It starts with everything's same, using either rabbit or mouse, and immunized with the various forms of antigens, DNA, purified proteins, as well as cells, VLPs, and we isolated and purified plasma B cells and used for single B cell sorting. And this becomes the critical point in what type of technology we choose to do so. At the Biointron, we call it AbDrop. This is a droplet, microdroplet based single B-cell screening platform in which we use an instrument that flows both the oil and the aqua phase from a different direction. And in the aqua phase, you have the B cells that's being flowed in a controlled speed, along with the reagent, along with the solutions that contains all the reagents needed for the bioassay, and the oil phase flows from a different direction. So it forms of micro droplets that contains only at the most one single cells with all the reagents inside. And after a short period of incubation time, the plasma B cell secretes antibodies, a tiny bit amount of antibodies, and if there's any binding with the antigen in the solution, using a FRET reaction, we can actually you can actually see a laser color shift and collect those positive droplets. So in this illustration, you can see in the droplet we have this plasma B cells, we have the antigens, so that's also labeled. We have a secondary antibody, and if it's binded, you have a FRET transfer, and it causes exciting of the lights at a different wavelength. After that, the droplets actually was collected, and then it's a further send it to microwells that is already contain barcoded primers, and the cells, the droplets are the cells are lysed, and the mRNAs are captured by the barcoded primers, and after a series of cDNA synthesis reaction, these cDNA, the mixture of the cDNAs can be sequenced using NGS, and because the primers are barcoded for either heavy chain or light chain, so the sequence data that we obtained contained paired sequence data for the light chain and the heavy chain that allow us to discover native antibody sequences. And the rest of that is relatively, it's Biointron's specialty technology, because we have been known for, well known in the field of antibody expression and production, and use our high throughput expression and the production system. We can mass produce these identified IgG sequences, so use the soup or purified form of antibodies for the binding assays or SPRs to validate the interaction with interagens. So within a week, within 1 week, the sequences are obtained, and in 2 weeks. So let me simply put this way. Okay, in 1 day, the AbDrop platform with the microfluid droplet system, we can sort millions of plasma B cells. And then after that, within a week, we can get an NGS sequence of thousands of clones. And then within a month, you can actually get the validated binders from this process. So the whole thing actually, in one time, you will get hundreds of naturally paired antibodies that's being validated. So this sounds already pretty good. However, we recently have formed a strategic partnership with a company called Cyagen, which is a leader in generating transgenic mice that has served the industry as well as academically for many years. And they have developed this fully humanized mouse called the HugoAb platform. And combining these two technologies, let me tell you how this works better, even better for your discovery process. The advantage of a humanized mouse is pretty obvious. So for all of us sitting here, It provides fully human antibody generation with the in vivo affinity maturation process in mouse. It greatly shortens the time for discovery process, and there's also potentially lower risk of immunogenicity because all the antibodies that we discover is human, and there's no need for humanization. So what is the HugoAb? The HugoAb is actually generated by Science's proprietary, they call the turbo knockout ES technology. This has an intellectual property of, this is the intellectual property of Science. They use a in situ process that replacing the complete VH-VK-VL with the human genes. And this mouse has been proven to have a robust immune response, which I will show you some of the data. And they are currently developing multiple mouse strains for different purposes. And the one that's currently available is called the HugoMab. The Hugo Light common light chain mouse is coming out very soon. But best of all, this platform is easily accessible with no milestones and the royalty that's commonly associated with many other humanized mouse platforms. And let me tell you a little bit more about the HugoMab mouse data. This slide showed you the B-cell development of a HugoMab. And you can see that the transition 1 and transition 2 B-cell development is very similar between the wild type and the HugoAb. And the mature B-cell level is also, I think it's quite similar. When we look at the marginal zone and the follicular B-cells, the marginal zone B-cells are very similar as well. The follicular B-cells are maybe slightly lower, but it's close enough. In the spleen, we look at the T-cell, B-cell, and NK cells, macrophage levels, and there's obviously no difference between the wild type and the HugoAb mouse. In terms of the IgG subtypes that these mouse generated, you can see when we compare to the native wild type versus HugoAb, the difference is very minimal. When we also compare the immunized wild type and the HugoAb, the response and the distribution of these subfamilies are also quite similar. So it's very similar to the wild type. And how about the usage of various germlines and the genes? And when we first look at the top panel for heavy chain gene distribution with the VDJ genes, we found that the distribution is actually quite similar that you could find in the human repertoire. And in particular, the CDR length is a very clear indication that in this figure, you can see this is the mouse CDR3 length distribution. And these two curves represents the human and the Hugo mouse MAB CD3 distribution, and they are very similar in my view. On the light chain compartment, the blue and the pink bar represents the comparison between wild type and the HugoMab mouse gene usage frequency. And you can see that the V gene, D gene, and the J gene frequency follows the same pattern as the wild type mouse. So on the next one, this is the light chain. Sorry, the previous one was the usage different subtypes of use. This is the slide for the light chain. We look at the light chain V genes and the J genes and the lambda VJ, and the distribution we found are very similar to you could find in the human repertoire. And we also did a comparison Hugo versus human subtypes, and their distribution is also very similar. So this is a good sign to see. Now I'd like to show you the immune response that we got from the Hugo mAb, which showed a pretty robust immune response compared to the wild type. Well, the disadvantage of this slide is that we actually don't see the wild type it doesn't separate with the wild type very well. But this is a good thing, right? So I highlighted, I circled the wild type mouse individuals, and they sort of blend in, and overall they are very similar. In some cases, you even see the wild type is slightly worse compared to the Hugo mAb mouse for some particular target. The immunization schemes are completely identical, so this is a head-to-head comparison between the two types of mice. On the next one, now this is the time that the Hugo mouse is combined with our microdroplet platform, AbDrop. And this is a head-to-head comparison using Hugo mouse comparing Hugo mouse and the wild type against the target PD-1 as a model system for discovery. And it follows the same immunization scheme, it follows the same method. And what we see is that these red dots are the microdroplets that's actually collected, sorted, and collected during the process. And this graph showed you the variance and the diversity distribution that we found in these two mouse, and we actually found more than 400 variants from the Hugo mouse versus 140 from the wild type. I don't necessarily consider the wild type is worse. I mean, there's a lot of variations there, but suffice to say that we could get a very good diversity from the Hugo mouse. The next, we also look at the binder characteristics, the binding characteristics of these sequences. So we picked 20 sequences from the sequence that we discovered, and look at the protein, the binding to PD-L1, and you can see that the EC50s are quite similar compared to there's a pretty good distribution and a very good EC50. And using atezolizumab as a reference, you can see that both mouse generated similar numbers of binders, similar numbers of EC50s. Some couple of them are even slightly better than the benchmark. The message is that these two actually really behaved quite similarly. And we also look at the cell binding of these sample sets, and found that 13 out of 15 from Hugo mAb versus 10 out of 17 from the wild type mouse showed significant binding to the cells expressing PD-L1. And among them, some of them are very similar to have a very similar EC50 compared to the benchmark. Not only that, we also look at the blocking activity of the subsets that we generated, and found that 6 versus 6, almost equal number, have the comparable blocking activity capability against PD-1 binding to PD-L1. So that's one case. The other case is also against a very popular target, VEGF. From the VEGF, we actually got close to 300 variants generated from Hugo-mab. A subset of that was expressed, and you can see that 13 out of 20 antibodies we produced showed very good binding affinity. That's sort of like a second case. The last case I'd like to show you is actually quite interesting because this is a direct comparison between AbDrop versus hybridoma. And the project background is like this. We have a project that we call Human Protein X because this is a client project. And so it shows a very high homology between the human and the mouse counterpart. And our customer, they actually used the hybridoma screening system to try to generate binders, but only got 2 of them. And then they turned to the AbDrop platform and using with the Hugo Ab mouse, and decided to hoping to get more binders. And indeed, the AbDrop platform generated, it sorted a total close to 1,000 droplets, positive droplets, and among them we identified 150 pairs of sequences. And from these sequences, we identified 34 unique pairs. That definition of unique is we have at least 2 amino acid differences among each other. So and the highest frequency showed up clone. It showed up 170 times, and so we actually expressed the entire set of 34 unique sequences and validated using the cell binding ELISA binding scheme. So experiment and see out of the 34, 28 of them showed the robust target binding activity. So I think this is a very good example of this system could actually generate more positive binders in some challenging situations. And when I simply put this, what's the advantage of joining force with Hugo Ab when we combine AbDrop? One is you got simply a human antibody. These are fully human genes in the in the mouse, and the time saved is time well earned because the whole thing takes about 3 months, considering maybe 6 to 8 weeks for immunization, and another month using AbDrop platform to discover validated antibodies. So this is much faster to get your human antibodies. And the high throughput allow us to dig deeper because 2 million B cells can be sorted and you get all the possibilities in this one single mouse. And best of all, like I said, the mouse platform comes with no string attached, no milestone and royalty payments with a one-time simple use access fee, which I think is very reasonable. So I hope in the coming year we will see more interesting cases that combining these 2 platforms to serve our customer better. And well, actually, Biointron, to many of you, you may heard of Biointron or even used Biointron for our 2-week antibody production services from the day you send us the sequence. This is what we are famous for, and we are this is also what we are, our focused business. And we now have more than 600 employees that serve more than 1,800 global customers. We are based in Shanghai, China, but we have an office in New Jersey. We have our scientists and staffs in the U.S. And our most popular RushMap service seems to be getting better. And simply put, from sequence to antibody takes only 2 weeks. And next year we're gonna provide more sophisticated packages with more data, including some of the initial screening in terms of developability and stability data that many of our customers would like to see when they use AI-generated or mass-generated sequences and wanted to see these data earlier. So the past year we have delivered more than 100,000 molecules, and we are looking forward to 2025 to serve more. So with that, thank you very much. We do have a booth at 409, and please find out more on our leading antibody expression services. And if then we also have our CEO here attending these conferences. And if you have any questions, you're welcome to ask us. Thank you so much. Okay.